I'd like to welcome our speaker, Dr. Nishi Rajakaruna. Nishi obtained his bachelor's in human ecology from the College of the Atlantic, but soon pivoted to studying plant ecology at the University of British Columbia, where he obtained his master's. He continued there for his PhD, where he studied the evolutionary ecology, Listenia californica, and Asteraceae. He then completed a postdoc at Stanford University, and started teaching botany at the College of the Atlantic, where he and then switched to studying at San Jose University and went back to teaching at College of the Atlantic. But ever since 2017, he's been a professor of plant biology at San Luis Obispo, where he's mentored many undergraduate and graduate students. He's also a Fulbright U.S. Scholar Alumni Ambassador. So if you have any questions about the Fulbright program, feel free to ask him during the reception after the seminar. His main research interests include examining the evolutionary and ecological consequences of plants interacting and adapting to harsh substrates. During the fall of 2024, I took on an independent study to assess the methods of soil analysis for plants occupying unusual soils, and Nishi's name kept popping up again and again over the papers I was reading, especially with the ones concerning serpentine. And so I know that, in the graduate program, we often talk about how important the substrate is for how plants interact with their environment. So I invited Nishi as a prominent researcher of geoecology to come speak today. The seminar has been about a year in the making, so I'm glad he's finally able to make it here. But it's been such a pleasure to have him in the garden already. So without further delay, I want to announce his presentation, Serpentine, Evolution and Ecology of a Model System. Everyone give him a big round of applause.
Thank you for that kind introduction. I'm sorry about my short lifespan wherever I work. I just looking for adventures all the time. So I keep moving from one job to another, but I've always have a great time wherever I am. So, and the same goes to being here. This has been like, Travis, I was just thinking, this kind of reminds me of going for a job interview. Like I've been meeting with so many of you guys, and the ideas are still so fresh in my mind from all of the exciting projects that the students are doing. And so I'm really, really happy to be here. And I'm glad I missed 2 lectures at Cal Poly to be here. I did send recorded lectures, but at the same time, it's been wonderful. It just got me excited about stuff as well. So as Charles pointed out, this meeting or this presentation has been in the making for a year. My father passed last year during the time that I was supposed to be here, so I had to move back to Sri Lanka for a while. But I'm glad you guys invited me back, and I was able to come back and talk to you. So my interests have been in geobotany from a very, very early age. I got excited about geobotany as an undergrad, even before I stepped on a serpentine outcrop. I got excited about serpentine, and I tell you how that happened. But so today's talk is really about like what we know about serpentine ecology and geobotany in general from the study of serpentine ecosystems. I'm going to emphasize a lot on information gaps and areas for future directions, hoping that some of you guys in the audience, the younger graduate students who got a career ahead of them, would take on some of these ideas and potentially keep me, be a collaborator of mine. And I've already worked with some of your former grad students. I just realized today that Nick Jensen was one of your grad students that I started a project with when he was a grad student. It's not quite done, so maybe one of you guys could take it on and finish it off. Before I get to my talk, I wanted to kind of talk to you about what I do at Cal Poly, because I was talking to Allison and talking about different lines of jobs in academia. Some people go to R1 universities, some people go to liberal arts colleges, some people go to like Cal Poly type places where it's like middle ground schools. I've been in all 3, right? And I consider myself a teacher. That is where I get most of my satisfaction. So I've loved working with undergrads. I teach a lot of different classes. When you're teaching focused universities, you get to learn botany all the time because you've been asked to teach stuff from like morphology and diversity to plant evolution and ecology. Currently, I only teach 2 classes, biogeography and ethnobotany. Every other year, I do teach geoecology, which is a class I taught in South Africa a couple of years ago that I've kind of turned it into a Cal Poly class. Before Cal Poly, I taught at College of the Atlantic, which is where I also did my undergrad. I was their very first foreign student, fresh out of the boat foreign student they had in 1990. It was quite the experience, a small school, 180 students at the time. So it kind of reminds me a little bit about what's going on here. Very strong like student-faculty interactions. a lot of community that you can see, feel in over here that I also felt as a foreign student at College of the Atlantic. In between College of the Atlantic and Cal Poly, I taught at San Jose State for 2 years as well. I was, in fact, the longest serving faculty in residence at Cal Poly, and I think at any CSU. So it was an extreme environment, and I always tell people that I was pre-adapted to extreme environments. I managed to live with first-year students in their dormitories for 4 years, did residential programs, evenings, weekend programs. Now I only teach part-time, but I've taken on a job as a coordinator or director of the new office that Cal Poly set up called National and International Fellowships and Scholarships. I help mostly undergrads and grad students, but I also help faculty and staff apply for competitive awards, especially for international travel and international research. I'll talk a little bit about that in a bit. I also advise clubs, and of course, this is my natural history club. I take students out on hikes every Sunday to teach local natural history. And because I come from Sri Lanka, I want to share a little bit of my British colonial heritage. So I have a tea club that meets every Tuesday night, and I also play cricket. It's probably the only CSU that has a cricket club that happens almost every weekend.
Nishi, what does that say after faculty?
Yaki Tutu, it's so with the First Nations land, so the Yaki Tutu names, so all of the Native American names are given to the residential communities at this new residential unit at Cal Poly. So this is the first year residential area. I don't think, did you stay there?
No.
No, you didn't, yeah. So just so you know, like I met Anika in my general botany class, and first time when she was a first year student. And I just realized today that I knew her from that class, but she tells me today that it was a Zoom class. So the fact that I got to know her pretty well over a Zoom class was pretty exciting to me that we still did. Yeah, anyways. So I am an ambassador for the US Fulbright Scholar Program. So I've been promoting Fulbright across the country among faculty, graduate students, and postdocs. And that's how I do my research. My research is international, and it's through networks of collaborators I've built through international travel. And I do encourage all of the graduate students here to think about the Fulbright program. The deadline is September 15th, October 7th, that time frame. It's a great time to go abroad. If you're finishing up your PhD or want to get a postdoc with someone that you want to work with abroad, it's fully covered. You can get research fellowships or teaching fellowships or teaching plus research fellowships. And it's pretty prestigious. If you get one, you're going to be embraced. You're going to be invited to the president's home in some of these countries because Fulbright is pretty well respected around the world. So if you are interested, let me know. I'm happy to take you through the application process. I did publish an article in the Plant Science Bulletin last year. It gives you detailed instructions on how to think about the Fulbright, how to get over that imposter syndrome of like, am I good enough for a Fulbright? Everyone can be good enough for a Fulbright. It's just a matter of going through that long application process. So read that. There's a PDF on my website too if you want to if you don't have access to the Plant Science Bulletin. And here I am at a site where we discovered a new genus of lichen. All the new discoveries I've done of lichens and bryophytes and other species have come through international collaborators done through Fulbright program. My first encounter with Welwitschia mirabilis in the Namib Desert was as part of a Fulbright opportunity that I had. It was with the Gyp World expedition. These are people from Spain and Turkey and Brazil and South Africa looking at some of these gypsum ecosystems in the southern Namibian region. And I don't know whether you know that Frederick Welwitsch, who actually the Austrian botanist who discovered Welwitschia, when he saw it, he knelt and wept in front of Welwitschia, and my reaction to Welwitschia was to actually jump over it because it was just this first year biology textbooks, you always read about Welwitschia, right? So to be able to see that, it would have not happened if not for a Fulbright award. So my research really has been about looking at mostly serpentine, but I'm interested in other systems as well. How do these island-like, sharply defined rock outcrop communities or habitats shape diversity, ecology, and evolution? I'm also interested in how to think about conservation policy and restoration practices in these island-like habitats that you get on land. I started working with plants, but now through collaborations, I work across different systems. I work with soil biocrust. I work with soil microfauna and mesofauna. Lichens is a big thing. I'm not a lichenologist, never taken a lichen class, but I've learned lichen through hanging out with lichenologists. So I get to do a lot of work on lichens on our substrates now, and I also work with bryophytes. And some of you guys actually know Laura Briscoe from New York Botanic Garden. She did a senior project with me at College of the Atlantic, and now is a curator at the New York Botanic Garden. I also work in different places, not just California. I'm interested in how this geology plant relationship works from different climatic systems, right? Because if you just work in California, you know how California systems work, like in terms of serpentine. But what happens in serpentine outcrops in California may not necessarily be what's happening in the tropical regions where there's high levels of rainfall. So saying things like serpentine soils are dry might not necessarily be applicable to a botanist in New Caledonia or Malaysia because it's rainforest, right? So I try to work across different systems, and for Fulbrights, I've worked at South Africa, Madagascar, Sri Lanka, and India. And because I worked at College of the Atlantic, we did some of the first kind of set of work on through undergraduate research projects, like with Laura Briscoe and Ian Medeiros. Yanni Brandwain, who's now a plant geneticist at plant evolutionary biologist at University of Minnesota, all did the senior projects with me on like mine tailings and serpentine outcrops up and down the East Coast. People often think serpentine is only found on California, but if you look across the Appalachian Belt, there's 600,000 hectares of serpentine on the East Coast. OMS and California has about 590,000 hectares. So actually there's more serpentine on the East Coast, but because these systems haven't had much time, ice-free time, to really kind of develop those floras that we have on the West Coast. So because the talk is on serpentine, but I want to kind of talk a little bit about why these systems are interesting, partly because they have really harsh soils. And what I mean by harsh soils is soils that have like too much or too little of essential nutrients, micro and macronutrients. They could be high or low pH, which makes pH actually determines nutrient availability and nutrient uptake. So high or low could be problematic. Some of these soils have high levels of toxic elements like metals and metalloids. Like for example, serpentine outcrops have high levels of nickel, chromium, cadmium, and they can be problematic for plant growth. Many of these things are in kind of exposed, rocky, slopes, and that generates like very shallow soils that have poor structure and texture. Some of them are too sandy, some of them could be too clayey, which could also be problematic in terms of nutrient uptake. And of course, all of them are bare, right? I mean, they're really exposed, not a lot of biomass growing in these outcrops. That becomes evapotranspiration, becomes a major problem for plants growing in these types of habitats. So while all of these things matter, I think of this as a kind of a harsh soil syndrome, like it's a synergistic edaphic kind of stressors that actually define what is able to grow and what is not able to grow. It's all of these interacting chemical, physical, and biological stressors because the soil biology shifts, right? I mean, as you go from a serpentine outcrop to a sandstone outcrop, the kinds of microbes change, soil mesofauna change. And so that is all of them working together is what creates the soil, makes the soil a harsh soil, right? It's very relative. What's harsh for one is not necessarily harsh for another. So one of the things that we see across all these multiple systems, whether it's working with gypsum or limestone or dolomite or iron or serpentine, generally there's a set of traits, functional traits that seems to be common among plants that are found in harsh soils. One of the key things I think is the low specific leaf area. I mean, some of the lowest specific leaf areas recorded for any plant currently comes from serpentine outcrops. They are tiny leaves. And if you look at gypsum, limestone, any of these soils that are exposed harsh have very low specific leaf area. This is leaf area divided by leaf mass, low growth rates, slow tissue turnover, low photosynthetic rates, often nutrient uptake rates. There's high investment in roots because you've got to access resources from resource poor soils. A lot of secondary metabolites, and this could be like in direct response to the stresses that they're dealing with. And another cool kind of trait that we see across these systems is that they have this really cool ion accumulation traits, either like halophytes taking up high levels of salt or gypsum plants taking high levels of sulfur, or the serpentine plants taking up high levels of heavy metals. So a lot of that stuff is put in the vacuum, and many of them, in place of some of the cations, could be used for osmotic adjustment to be able to take water from water-limited systems. So the modern systems that have developed right now that are, there's a lot of work going on serpentine. Of course, there's a long history of serpentine geoecology happening in California from the early 1940s to the current time. Gypsum is pretty big in Spain. There's a lot of people working with gypsum systems, and there's a lot of parallels, even though the soils are different. One is high calcium, one is very low calcium. but there are similar kind of responses to these soils across these calcium-rich and calcium-poor substrates. And Michael Moore, who happened to be Charles's undergraduate advisor that I found out, also works on gypsum. He's at Oberlin College where he did his undergrad. There are other systems, of course, saline systems have a long history of research in terms of understanding soil tolerance, dolomite, volcanic soils, guano deposit, Lasinia maritima, which is a plant that Mike Wasey worked with Onda a long time ago, is a guano endemic, right? It's close relative is Lasinia minor, which is a coastal bluff species. So that guano tolerance, you've got to deal with, it's the opposite problem, like too much nutrients, there's too much phosphorus, too much nitrogen. How do you deal with that? So one of the common themes that we see across these systems is that these habitats, because of the stress associated with these habitats, there's not a lot of biomass. The habitats are bare, which means that you have to deal with high levels of evapotranspiration, hold on to water as much as possible, right? So this is the project that I'm looking for a collaborator with that actually started this with Nick Jensen when he was a graduate student here. I actually didn't remember that until I started talking to Travis and others during lunch today. One of the projects that I've been interested in is trying to kind of really show like how much of an influence geology has played in generating California's flora in terms of endemism. The work that Nick and I did when he was a grad student here showed that about 50% of California's endemic plants qualify as harsh soil specialists. Like they could be iron, they could be shale, they could be sandstone, they could be serpentine, they could be some other substrate. But if you look at the CCH records and talking to people, it seems like 75% of the time was sort of cut off, they're found in a harsh soil. And what was interesting to us at the time, this could have changed since the time I started working on this project, about half of them were also CRPR ranked plants. So not only are they harsh soils are playing an important role in terms of endemism in California, but most of those endemics also happen to be rare, right? So this kind of makes you pay attention to soil plant substrate associations or plant substrate associations. Because if you're thinking about any kind of conservation plan or a restoration effort, right, or you're developing species distribution models, if you don't have substrate associations, you're not going to come up with, good, conclusions in terms of how you want to approach this project. So this goes with all of the climate change models that are coming out from David Ackley's lab, who was my postdoctoral advisor. None of these models have substrate associated with predicting how plants are going to move with response to climate change. You cannot just move up a slope or move north if you are a substrate endemic. It depends on how quickly and how far and how effectively you can disturb from one island to another island, right? That could be, maybe 100 kilometers away, right? So the whole conservation planning idea with respect to climate change will only be better informed if we incorporate soil layers into climate change models. I'm going to talk about serpentine, and I'll try to stay on topic so that I don't get distracted too much. One of the things I do think would be useful for you guys, except perhaps Chloe, who seems to be like this amazing geobotany I just ran into a little while ago. She loves rocks, she loves plants. Is like a lot of botanists tell me, like most botanists don't take soil science, right? They don't take geology, they have no idea anything about rocks. I learned rocks and soils partly because of the work that I'm interested in, but never took it as an undergrad or a grad student. But one of the coolest things about knowing where you're on a serpentine outcrop is to carry a Harry Potter wand or a magnetic pen that you can buy for $5 on Amazon. Serpentinite has magnetite, all ultramafic rocks has magnetite. So the rocks have a magnetic pull. So if you're a botanist who don't know the difference between serpentine and a sandstone or shale or some other adjacent rock type, as soon as you start picking up rock with your extendable magnet, you don't have to bend down. As you get older, it's harder, right? So it's easy to pick things up with your magnet. And you are on an ultramafic body, right? You have high iron magnesium rock. So something to carry around in your toolbox when you're wandering around. So serpentine, you guys have already heard about it. This is your state rock. Everybody should have a rock at home. You should be carrying it in your car. You should have a bumper sticker. If you want a bumper sticker, reach out to me. I have 2 bumper stickers. One is called I break for serpentine. The other one says serpentine rocks, exclamation point. If you want a bumper sticker, let me know. I'm happy to mail it to you. So these are rocks that have high levels of iron and magnesium. They're mantle rocks. They are found on continental margins, in subduction zones, fault lines, mountain belts. Very, very high levels of nickel and chromium. The ones in red are like the key things that you want to pay attention to. High levels of heavy metal. except nickel and manganese, the others are really not essential, like they're not micronutrients. The problem is the calcium to magnesium ratio, which is universal, like very low levels of calcium, because when serpentinite rock is formed, calcium is leached out, right? And it gets replaced by magnesium, which is another divalent cation. So there's absolutely very little calcium. Most serpentine soils in California has less than 0.7 calcium to magnesium ratio, right? And this is consistent even in tropical landscapes. Once you get to the soil just above the bedrock, the soils do show this calcium to magnesium ratio. Right? Nutrients can change. When you say that serpentine soils are nutrient poor to a bunch of tropical botanists, they laugh at you, right? They're not dry, they are wet, there's high levels of organic matter, there's high levels of nutrients. So the nutrient thing matters for California and Mediterranean systems, but not necessarily tropical systems. A lot of geobotany is dominated by Krukerberg's ideas. And Krukerberg obviously worked in California. So now with all these other international people coming in, people are trying to kind of say, hey, let's take a step back. We've got to think about these systems with respect to climate. It really matters. Right? So generally, in most places in Mediterranean climates, you have barren settings like New Idria. I know that Travis has been there, Charles has been there, a beautiful moonscape. In 1951, Krukerberg wrote his in diary, have I landed on the far side of the moon, all soils and rocks, no plants here. Right? This was his first kind of impression of in New Idria when he went there in 1951. Distinct plant communities, because of these harsh conditions, you can think of them islands on land, right? So while we are excited about serpentine, microbiologists and NASA scientists, astrobiologists are interested in serpentine because there's this idea that life began in places where serpentinization was happening, right? Because the whole conversion of mantle rock to serpentinite rock involves a kind of energy, like a lot of energy that's been built, and raw materials like hydrogen sulfide, and methane in large quantities that are built, which they think would have supported the early chemosynthetic bacteria, right? So there's a ton of work going on, like the mid-oceanic ridges, and in places where they think serpentinizing is happening in surface areas, where they find microbes that kind of represent what we think of as an early microbe that would have kind of evolved in this kind of soap that was generated in terms of generating life. So there's a ton of papers. I mean, if you type in serpentinization and early life, there's a lot of work in this area. A lot of work is done in Europe, but Stanford has some research going on. NASA has researchers doing this work across the world. Rather than going underwater, you can actually study this in places like Cedars, which also is a place that Travis has been to. Chloe, have you been here?
Yeah.
It's a fantastic spot. It's like Mars. It's like red rocks, mostly peridotite. What's interesting is it's like one of the few places in the world where that whole process of serpentinization is happening on the surface. So you see these like serpentinite rocks, peridotite, which is a mantle rock, and right next to it, you get these pools of calcium carbonate, which was just leached out during that process. So it's a really cool setting to look at like a gradient of serpentine chemistry from no calcium to really high calcium to see how plants might respond to that kind of environment, and no work has been done on that area yet. Serpentine is important in your life, right? Asbestos, which we may have, like Lucinda and I, and maybe Travis used to inhale these things accidentally not knowing, but back in the day, we had these things in buildings, right? Stainless steel, nickel, and chromium would not be around. You wouldn't have a stainless steel spoon if not for mining serpentinite rocks around the world. If you go to places like Cuba and South Africa, you see people selling jewelry made out of serpentine. Very easy rock to work with. But this is the big issue right now, right? This is electric vehicles coming in, and the nickel demand is going up. Huge amounts of nickel is going to be needed if you want to keep this electrical vehicle thing going on. And the only way this can be done is if there's extensive mining happening in countries where there's large, extensive serpentine outcrops. Right? So nickel mining is going to be a big problem in terms of serpentine conservation issues down the line, and something to think about with respect to like future stresses associated with these environments. So I, fell in love with serpentine at a very young age, as a second year undergrad. Back in the day, there's no internet. You go into your advisor's filing cabinet, you pull out a paper, and I pulled out this chapter, Plant Life on Western North America by Krukerberg, read that, and then managed to read the essay, The Stimulus of Unusual Geology, so plant appreciation. I was hooked. Right? I'd never seen a serpentine outcrop. Didn't know who Krukerberg was, but his writing just got me. And then, of course, got to know his work over time, and I'm sure you guys have seen some of these books. I mean, if you're interested in geobotany, I would recommend you pull out, get this book and at least this one. The Geology and Plant Life is another book, the last book he wrote. He didn't write a lot of papers, but his papers were very important in terms of shaping the field of evolutionary ecology, and I'll talk a little bit about how he shaped the field over time. But I, again, had the privilege and the honor of being asked by his kids to write his in memoriam, which I wrote in 2016, because I got to know Krukerberg later in life, and I spent a lot of time with him, and just a fantastic person. So if you're interested in learning about my relationship with Krukerberg, here's an article to read. But in terms of Krukerberg's contributions to botany or ecology, Brian and Acker's paper that came in AJB during the Centennial Review issue, in the Centennial Review issue, kind of highlights the way that Krukerberg's first paper on intraspecific variation in serpentine plants and using of like reciprocal transplant experiment and common garden studies shaped our understanding of kind of the plant evolutionary process, including like local adaptation and functional traits, speciation process, as well as the importance of climate and soil in shaping diversity, which is what we are still doing, right? When the foundation of that was done by Krugerberg. Great paper to read if you're interested in kind of looking at how some of the early literature shaped what we do today in the name of evolutionary ecology. Clearly inspired my research. I was on the East Coast working in a plant ecophysiology lab, but serpentine was in the back of my mind, and I wanted to get to the West Coast because at the time I thought that was the only place serpentine was. Krugerberg was not accepting students at the time. I actually ended up at UBC working with a phytochemist who was good friends with Krugerberg, so I actually had access to Krugerberg through Bruce Bohm. And my first paper was really kind of describing this cool phytochemical pattern that was seen in Leycinia californica complex, or Leycinia californica at the time, where there were two chemical racers. One was called race C /B. B dropped off along the way, I don't know why, and then there was race A. And the two racers were found on the serpentine outcrop at Jasper Ridge at Stanford. And year after year, they would maintain this very sharp boundary. Like they would be found at the very bottom of the sail or the top of the swale. And there, this line would shift by maybe half a meter year to year over the 5 years I looked at that population. Prior to me, my advice had been looking at this population for like 10, 15 years, right? And actually, this is Robert Onduff right here. I wasn't there when this photo was taken, but Robert Onduff and Bruce's wife, Leslie, who's up there. So what we showed was that these chemical races or ecotypes that was found at Jasper Ridge was found across the state. And they differed by their flavonoid chemistry, and one had this really cool chemistry type that is found mostly in plants that are found in salt environments. I was talking to Erica about her work with Orobanki, and there is this sulfate diglycoside that seems to be very common among plants that are found in ionically extreme soils. And I found the same compound in this race that's found here. There was some where there's allozyme differences, and at the same, allozymes that nobody does anymore, but back in the day, we used allozymes to look at population structure. And there were some subtle differences in actin morphology, and flowering times are different. You can actually see this in the photo. These guys flower about 7 to 10 days after these guys flower, right? What was interesting is that when we did reciprocal transplant and common garden studies, we showed that the two races were locally adapted where they're found. Reciprocal transplants, these guys don't make it here, they're too dry for them, they just have high levels of mortality. These guys cannot tolerate the soil here. It's too ionically extreme, very, very high pH, high levels of sodium, which is leaching down from the upper slopes of the outcrop to the bottom swale. We also did greenhouse work, hydroponic studies, and breeding studies in the greenhouse that showed that there's higher fitness among race C under drought. In fact, they're able to maintain high levels of reproductive fitness across the kind of water stress gradient, whereas these guys they need to be growing in water. They are found in more wetter serpentine soils. And they also have really highly efficient sodium magnesium transport systems. They take up these ions in really, really high numbers. And because they are growing in ionically extreme soils, we were suggesting at the time that they may be using sodium and magnesium as a way to kind of osmotically adjust themselves to take up water from ionically extreme soil, just like halophytes do. Right? What was really cool about this is that the race A has the same compound that a lot of the halophytes have in terms of the flavonoid chemistry. We did breeding studies, which led me, I was telling who works on Asteraceae erycomeria, Samantha, I was telling her to be very careful with Asteraceae. If you work with Asteraceae for too long, you're going to develop severe allergies, and you don't want to be allergic to one of the largest plant families in the world, which kind of really ruins your career as a botanist. So I was not over that. Bruce Baldwin has issues. Bruce Bohm has issues. People who work with Asteraceae for a long period of time develop severe allergies. You can get all the secondary chemicals going through your skin. You inhale it. So be careful when you're working with Asteraceae, especially when you're doing like pollination studies like I did. Right? We showed that they have very low reproductive compatibility as well. This is not, so now I'm ready to write this thing. Like a month before I'm supposed to start writing, a paper comes out that shows that Leycinia californica, as I knew it, is not monophyletic, and that they resurrected an old species called Leycinia gracilis and said, oh, there are two separate clades. There's a southern clade and a northern clade. The northern clade will be Leycinia californica. The southern clade will be gracilis. So I was going to kind of describe this as a kind of a local speciation event, or like two ecotypes found in different habitats. They're locally fit to this. So it's now like, how do my races fit in with the species that have been described, right? So this caused some stress, like just finishing up your PhD, you don't have funding now, but then somebody's saying that, how do your races correspond to the species that we know, right? So, but what's interesting is that when we sequence the races and we sent the races to Berkeley, Bruce Baldwin's lab did the work, and we showed that the races are found in both these species. It really depends on where they are growing. Whether it's Californica, gracilis, it doesn't matter. If you're growing in a harsh soil environment like a vernal pool or a coastal bluff or a serpentine outcrop bottom swale, you're more likely to be race A, right? It has a flavonoid profile of race A. So this is how I ended up finishing my saying and kind of leading with the question. So, and what was really cool about Jasper Ridge is that race A was Californica and race C was gracilis. So the northern and the southern species of meeting at Jasper Ridge, and they happened to be two races, but they also happened to be from the two separate clades meeting on this one outcrop, right? Which was a really cool setting. So this ended up me proposing this as a model for parallel evolution, right? We have two separate close related clades. They both seem to have these kind of similar ecotypes or edaphic races. So this is where it's at, a 20-year-old hypothesis that has not been looked at. And I think it's easy to grow plant. This is across California. We know the flavonoid profiles of most of these populations. Because I moved to Maine and got stuck on a little island teaching amazing undergrads botany for 14 years, I never could pursue some of the work that I had started. So this is a cool project for anybody who's interested in doing this. There's cool methods out there to figure this out, but kind of proposing this as a model for parallel evolution. Serpentine is, you think serpentine is everywhere, but it's only 1% of the Earth's covered by serpentine. But what's interesting, if you look at the biodiversity hotspots, you find that a lot of these outcrops actually are also found on some of the biodiversity hotspots. If you impose other types of geologies, you find a very strong correspondence of harsh substrates associated with biodiversity hotspots. So they play a big role in terms of biodiversity. Right. So for people, I just was talking to Provi, like there's a conference that happens every 2 to 3 years on serpentine ecology. There have been 11 so far. The first one was at UC Davis, where they honored Krukerberg and a couple of other scientists as kind of founders of this field. I started attending in 1991, so I'm one of the few living people, strangely enough. I still feel pretty young, but there are only 3 people in the world who've attended more than 9 conferences right now, because everyone else has passed on. And I did organize this conference in all places in Maine, which is not the most exciting botanically, but we went to Mount Albert in Gaspé Peninsula, which is kind of very high alpine serpentines, which is really cool. There is an allotetraploid fern, which is endemic to serpentine in the East Coast. It's an Adiantum viridimontanum, which is a hybrid between Adiantum pedatum, which is a forest fern, and Adiantum aleuticum, which is also here. There's a cool disjunction. There are some serpentine endemic ferns on the East Coast. They're largely serpentine endemic. On the West Coast, they're bourgeois. They're on and off, like aleuticum, right? And I think it has something to do with rainfall. But there are some, like Aspidosis densa is another one. It's largely endemic to serpentine on the East Coast, but on and off of serpentine on the West Coast. So the first conference was to honor Krukeberg, Robert Brooks, who wrote a lot about serpentine botany all over the world. He was mostly a phytochemist interested in heavy metal uptake. And then she did a lot of work on ion uptake, especially heavy metal accumulation, and she's from Italy. So it was really honoring these three founders of the field. The last one was in Kyoto conference, Kyoto, Japan. I took 6 Cal Poly students with me. It was really great. There were people from 43 countries, and there were about 150 people at the conference. It happens every 3 years. But the next one is actually going to be in Serbia. If anybody is interested in geobotany or any aspect of geobotany, do get in touch with me. I'll send you the registration details. The conference website will go up in a couple more months, and it's an opportunity to go see Eastern Europe if you've never been to that part of the world. All of these conferences are a great way for undergrads to get publications. So a lot of my undergrads actually published their first paper in the conference proceedings, which is kind of a way to get them started publishing their work. So in terms of hotspots, there's serpentine everywhere along continental margin, but New Caledonia, I realized that Carl Quist spent a lot of time working in New Caledonia today. I didn't realize that part of his life until Anika took me to the herbarium this morning. But 83% of New Caledonian endemics are serpentine endemics. They're monotypic genera on serpentine. Cuba has a lot, right? Both New Caledonia and Cuba are devastated by nickel and chromium mining. Just so you know, it's just a sad scene to see. But they also happen to be highly biodiverse regions, partly because of the serpentine bedrock. Malaysia is an up-and-coming area, Sabah, island of Borneo. So far, we think there's at least 5% total vascular plants endemic to serpentine soils, including these amazing pitcher plants, like Nepenthes species, that are largely restricted. I wouldn't say they're 100%, but I would say over 80% of the time, these pitcher plants are avoiding granite and gneiss. They're found mostly on the ultramafic sides of the Mount Kinabalu. And then this is for Allison. There are some really cool orchids out there that is also largely restricted to serpentine landscapes in Malaysia. These are not my photos. These are photos of my collaborator, Anthony van der End, who spends a lot of time in Malaysia working on serpentine plants. California is interesting. 15% of California endemics are serpentine endemics. And Hugh Safford and Jesse Miller, I'm sure you guys know these people, have been maintaining this really cool database looking at serpentine affinity across California. So even though 1.5% of the California's landscape has exposed serpentine, about 15% of our plants are endemic to serpentine soils, right? And this database is pretty cool. It was first written in 2005, updated in 2020. It gives you kind of a range, right? Because there's nothing's 100%, but it gives you like over 90% is a strict endemic, over 85% is a broad endemic, all the way to like below 50%, where it's like a boarding bog or something that is able to tolerate serpentine. It gives us an idea about how serpentine plants in California is associated with this unique substrate. But what I want to point out is that while this database trends hold broadly across California, what is interesting to me is how those exceptions, right? There are times you see plants that they don't call a serpentine strict endemic, and suddenly you're in San Luis Obispo on a south-facing slope, and that's the only place you find this plant is south, and it's an endemic to south-facing slopes in one part of California, and is broadly found in other places. So there are these exceptions that I think generate a lot of interesting, questions in terms of how climate and maybe land use history or other factors might play in terms of in terms of restriction of certain plants, right? So, but statewide, this is a great database to go to if you want to know whether your plant is endemic to serpentine or not. So, for example, I was just talking to Allison, and both these plants came up. These are some of my favorite serpentine endemics. This is a broad endemic, Cypripedium californicum. They're on serpentine fence. So it's a bedrock, serpentinite or peridotite, and then you have a bog-like setting, contact with groundwater, and you have Darlingtonia, Rhododendron occidentalis, and Cypripedium californicum growing in. If you have never seen a Darlingtonia, you guys are doing field work, all of you guys, so you would have seen it. And I teach botany at Cal Poly, like, no one's seen a Darlingtonia. I'm like, you grew up in California, you've got to go to Northern California and say hi to a Darlingtonia. So even in San Luis Obispo, given it's like Central California, you don't expect a lot of endemism in like drier parts of California, but we do have quite a number of endemics in San Luis Obispo. Back in the day, I did not do this trip to Serpentine. Sometimes when the California Botanical Society comes, I take students to Irish Hills to show some of these cool plants. I think my grad student, Mike Butler, took you guys with Dina Grossenbacher last meeting. I was in Sri Lanka at the time. But there's some really cool plants even in San Luis Obispo, highly endemic to serpentine soils. New endemics are being discovered and reported, and I was talking to Travis about the fact that people don't put chromosome numbers anymore, right? There's no genetic information, but based on morphologies, things have been described. I don't really know whether these are like true species or like phenotypic variation or local adapted types, but these have been described as new species in recent times. There was a fish species which happens to be the world's first endemic to Serpentine described, the Red Hills roach. It's only found in pools that are on top of serpentinite rocks, and it's very locally restricted fish species that was described in 2019 as the world's first serpentine endemic vertebrate, right? So I mean, California is pretty amazing in terms of like finding all these like geobotanical or geological wonders in terms of biology. So Susan Harrison has been doing a lot of cool work in terms of statewide patterns, showing that higher precipitation, which you get in northern climes like Northern California, Klamath Mountains, have higher levels of endemism. So the ideas behind that currently are that higher precipitation levels drive greater regional biomass, high competition, so stress-tolerant species could get restricted to these island-like habitats. Also, if you go to Northern California, there are older outcrops that have much, much more extensive landscapes covered, a lot of microhabitats, heterogeneity landscape-wise might provide those opportunities for radiations to occur. So generally, higher endemism in Northern California than in Southern California, and it's almost like an inverse latitudinal gradient, right? And if you think about generally going south towards the equator, you find greater levels of species. But in terms of serpentine endemism, you find like the inverse of that happening across California. Brian Onacker did some really cool work. It's too bad he's not in academia. I really liked it. I worked with him when he was a grad student with Susan Harrison. But he did this kind of meta-analysis of phylogenies that were available for California plants that had serpentine-tolerant or endemic species, and showed that when you look at the kind of the directionality of evolution for serpentine tolerance, that most of the time you have endemics evolving either directly from non-tolerant ancestors or from tolerant ancestors, right? That were like on and off of serpentine, and then their sister taxon or their descendant taxon is an endemic to serpentine. So generally the directionality is towards endemism, and this is based on, I think, 23 genera, 708 taxa, 51 serpentine endemics within this genera. I mean, now there are way more phylogenies available since like 2008-9 when he did this work that has serpentine taxa included. So updating this thing to show whether the same pattern is there, it will be really cool. He did document some reversals from endemism to tolerator, and one instance in the Brassicaceae, I think in the Streptanthus genus, where there was a reversal of an endemic giving rise to a non-tolerator. So the arrow thickness kind of represent the frequency of the observed transition that he found. And this kind of begs the question of whether the becoming an endemic is an evolutionary dead end. Like there's no further diversification in those clades that have high levels of serpentine endemics, right? But again, like we have way more genera now with phylogenies available. It'd be a really cool test to see whether this pattern holds across California now. The reason why some species become endemics and others don't maintain populations on and off, or not or tolerant but never endemic, is something that's needs to be tested more. There are a couple of ideas as to why plants may become endemic. One is that the high cost to tolerance associated with serpentine might kind of lead to that restriction. So there's a competitive trade-off hypothesis that endemics are not as tolerant to serpentine compared to regular plants. So because you're not as competitive, you get restricted. And then the other one is the defense trade-off hypothesis, the idea that increased susceptibility to herbivory among serpentine endemics of serpentine soil, and that might also push them to be more restricted to serpentine soil. So these are the current ongoing hypotheses as to why serpentine plants maybe endemics are on serpentine, but targeted experiments are needed to test this competitive versus defense trade-off hypothesis. There's some work done by Kathleen Kay's lab, but there's a lot more that needs to be done to really figure out what's going on. And different lineages might be exposed to different reasons as to why this is happening. Overall, though, the endemics are found in soils that are harsher, right? So endemics are in soils that have low calcium, much lower than their tolerant and cousins grow on. They also happen to be on more barren soils. So general tendency across Mediterranean climates. This is for California, but there's other work done in Spain and Italy and Greece that also shows that endemics are found in more bare, harsher substrates. And the work that Sharon Strauss has done shows that in a lot in that this is Strepcanthus group, that being able to tolerate bare habitat seems to be precursor for serpentine endemism. So a lot of the ancestors of serpentine endemics happen to be plants that are found in bare soils, right? So they're already pre-adapted to dealing with UV radiation and all of these other stresses that are associated with an open habitat. That it's like the next step forward in the evolution process. One of the things that have been interested me for a while is this idea that if you're in a barren habitat, you're obviously going to be seen more, right? So Sharon Strauss and Ivalu Kako did this paper that was published in American Naturalist that basically kind of looks at whether serpentine plants are better camouflaged in serpentine soils with this better color matching of foliage with the substrate color. And this is the kind of work that I think could be done more of. We don't really know much whether this is happening in other clades, but there seems to be better color matching of endemics on serpentine soils than their closest ancestors of serpentine. And at the same time, Stephanie Porter did work in 2013 looking at seed color matching, right? So the endemics seem to have seeds that better match with the serpentine substrate than their closest relatives that were off serpentine. Right? So that could be an interesting adaptation that is understudied, so to speak, that could be a kind of a great avenue for research in the future. One of the things we know is that serpentine plants have smaller leaves. It doesn't matter whether you're growing in a high rainfall area, whether you're growing in New Caledonia, or whether you're growing in California or up and down California, they will always have lower specific leaf area. They happen to be in terms of the specific leaf area database that's out there, serpentine plants are the ones that have the lowest ones across a global database. Right? So, it seems like they don't really care about the climatic precipitation component. Like they have lower specific leaf area, primarily because they're dealing with like soils that are harsh, nutrient uptake is difficult, either nutrients are lacking. So they may be more constrained by nutrients or lack of ability to take up nutrients, and that's why they have smaller leaves rather than having water being the driving force between specific leaf area. Here's a paper we did with collaborators from across the world who basically collected the 50 most dominant plants on serpentine and the adjacent plants of serpentine, and we reported that in all these three areas, the serpentine plants had the lowest specific leaf. It was a kind of a first round to see whether this is actually happening across multiple families in multiple regions. Another trait that I think would be cool, given you're interested in traits, one of the things that I've noticed in Sri Lanka is that evolvulus alsinoides in the Convolvulaceae, when growing on serpentine soils, have pink flowers, and then off serpentine, they have white flowers. Doug Shemsky reported this with Leptosiphon, back in the day it was called Lionanthus. Calincia, Maureen Stanton and Yoni Moyle at UC Davis did this work with Jessica Wright, showing that there's this flower color difference happening. There's great anthocyanin content in plants that are on serpentine in several lineages. And Dina Grossenbacher and group did a paper recently that shows that climate is obviously precipitation is contributing to this pattern. But again, whether there are fitness trade-off in contrasting habitats, we don't know anything about how flower colors play a role in terms of soil restriction, and whether the pigments themselves confer tolerance to the stresses that they're dealing with. While endemism, I'm going to wrap this up very quickly. While there's endemism in terms of flowering plants, one of the things that we don't see is high levels of endemism among cryptogams. There are no endemics, even though Jim Sherlock described one moss in Northern California that appears to be endemic to serpentine. There are times that these so-called endemics appear on other types of rocks later, right? It's just a matter of not finding them. We've done quite a bit of work with lichens, and we're not seeing that pattern. The pattern that we do see is that there are distinct communities. You can have a sandstone rock or a shale rock or a granite rock and serpentine like 10 meters away. The communities are different, right? So there is community assembly patterns that differ between these rock types, and there are regional indicators of serpentine across the world. Like you can go to a part of the world and say these 6 lichens found are most likely going to indicate a serpentinite rock. But they will be found on other type of rocks, right? So they're not as restricted as vascular plants is, right? And this is having anything to do with the ion optics. I mean, the fact that they're not actively taking up ions, they're like non-vascular, whether that has anything to do with their broader tolerance of substrate level stuff, elements, it's unclear, but there's no pattern similar. Whereas with mesofauna and microfauna, we again find a similar pattern to these guys. There are distinct communities, even at Irish Hills. You go to Serpentine outcrop and you sample, springtails and mites and nematodes on serpentine and sandstone, you find very different communities. None of them are endemic, but they're distinct across these substrates. Right? I don't know whether you've seen this book. This is a book that Susan Harrison and I wrote in 2011. There are 9 chapters on Earth history and evolution. So this is like, what can serpentine teach us about ecology, evolution, and conservation biology? I mean, this book was really meant for non-serpentine biologists, right? To say, hey, why you can use serpentine as a model system to ask questions in ecology and evolutionary biology you are interested in, that you've been working on. Like, think of this as a model system. Okay? And I'm not going to have time to talk about spatial ecology. This is 30 years of work that Susan Harrison has done on looking at kind of community ecology, meta-population ideas, island biogeography theory as ways of incorporating, using serpentine as a model system to ask these questions. So this paper, which is not available widely, she was asked to write this paper for some Finnish journal. And she writes about her 30 years of experience. If you want a PDF of this book, I mean, this is a great chapter. Like it's really kind of brings together all of these macroecology ideas and how serpentine could be a great model system or not to ask some of these macroecological questions. In terms of evolutionary processes, which is where my journey started, very, very clearly, these are great model systems to ask questions about evolution across adjacent soil types. These ecotypes that we often find are reproductively isolated through flowering time differences and other types of traits. Quickly, we know that calcium to magnesium ion homeostasis is really important in terms of lineages. We have looked at heavy metal detoxification, and drought tolerance seems to be like the key 3 areas across multiple families that seem to be critical in terms of like genetic differences across lineages that are serpentine tolerant or not or serpentine intolerant. There are lots of examples of traits that have shifted with respect to reproductive isolating traits. There are pollinator shifts, flowering time differences, hybrid sterility. There's a lot of work in this area that shows that some of these traits actually might also be connected. So some of the traits that are responsible for local adaptation or adaptation might be linked with traits that are responsible for reproductive isolation. Also, so therefore, through pleiotropy or linkage, some of these genes could be acting together to almost bring about instant reproductive isolation in response to an adaptation to a soil condition. Here's a good cool paper you can look at. Czech Republic folks have been working on the genomics of serpentine adaptation, looking at the kind of the links between approaches to studying links between genotype, phenotype, and fitness in serpentine environment. So this paper review that came up in Frontiers in Plant Science looks at kind of how to bring these ideas together in terms of looking at adaptations and reproductive isolation traits and fitness in serpentine soils. This is a topic that I try to finish in 2 minutes, and then I'll give you guys 5 minutes to talk about. One of the cool functional traits that is not talked about often among serpentine adaptation is this idea that, or this observation we made, that some plants take up really huge amounts of heavy metals into their tissue. A lot of these heavy metals end up in the trichomes, in the latex, in the phloem sap, or in the upper or lower epidermis of a leaf. So they are very clear about where they put these heavy metals. They are often chelated. There are polypeptides and organic compounds that are expressed right at the point that the ion enters the xylem stream, and then they are translocated as a chelated compound to a vacuole. And now with all these cool micro X-ray techniques we have, we can also look at like anthers and nectaries and ovary walls. And this shows you different elemental profiles in flower pods, showing that a lot of the heavy metals are in the anthers, ovary wall, ovules, and in the nectaries. Nectaries actually have a lot of heavy metals, even in plants like Chaptanthus polygonioides, right? So there's differential translocation, localization of heavy metals. And this is a really up and coming technique that's out there. People are doing this kind of X-ray fluorescence microscopy to look at elemental profiles in tissues. And when I was in the good anatomy lab, I thought, like, you guys have SCM, like this is the kind of thing that we could potentially do with some of the serpentine plants that we know are taking up heavy metals, right? So first plant was discovered in 1975, published in Science. This takes up 25% of its dry weight in nickel. This 1% could put us in the hospital, right? I mean, like 25% is pretty bad. It mostly is in the latex, but all parts of the plant tissue has high levels of heavy metals that are beyond the threshold for hyperaccumulation, which is 0.1%. And right now, there are about 700 species from serpentine outcrops that have been described as hyperaccumulating metals or metalloids or rare earth elements, but 500 of them happen to be taking up nickel. So nickel seems to be the most widely uptaken element. It is very similar in size to magnesium and calcium, so it tends to be probably outcompeting magnesium channels when there's high levels of nickel and low levels of calcium, but it also gets in through zinc transport systems. Why plants take up heavy metals is something that evolutionary biologists have been interested in, and this is an understudied area. One of the hypotheses is metal defense, that plants take up heavy metals, put them in like phloem, which is sugar-rich, latex, which is a defense strategy compound, epidermis, which is the first point of contact, trichomes, where things would bite you and a trichome would break and release the nickel. So it should deter things that are eating you. But of course, there's really cool co-evolutionary opportunities for studies. There are insects that are monophagous on metal hyperaccumulating plants. This is on Burkia codi from Asteraceae in South Africa. This is your claim to fame, Melanotrichus boydii, which is your monophagous insect that is only feeding on Streptanthus polygloides, which is one of two California's nickel hyperaccumulating plants found in the foothills, Sierra foothills. And some of them take up high levels of nickel, so they could be point of contacts to move metals out of the serpentine ecosystem, so trophic transfer. Some of them actually pass it through their gut, so they end up in the scap right away. So there are different mechanisms in the insects that deal with these heavy metals. Right now, around the world, there are 17 insects that are termed nickel insects because they are feeding on nickel hyperaccumulating plants, and that's all they feed on. Elemental allelopathy is another hypothesis that's out there. The idea that these leaves that have heavy metals drop underneath the tree, then enrich the heavy metal underneath the canopy, that might then keep other non-tolerant plants from invading your space. There's some research suggesting that might be happening, and especially that study from New Caledonia shows that it happens. Drought tolerance is something that I would love to have people test. There hasn't really been a lot of tests on it to show whether these divalent cations like nickel can be used in a potassium deficient environment. Potassium is what's usually used for osmotic adjustment. There's very little potassium in serpentine soils. So if you are able to take up nickel as a divalent cation, are you using this as a way to osmotically adjust yourself to be able to take water from water-limiting soils? So Molly Warrington, who was one of my undergraduate students who took my geology class, did his final project on this, and we finally managed to get this actually published in ecological research that's coming out. We've proposed 7 hypotheses. If you're interested in this, because there's so much opportunity for research, understanding why plants do this weird thing, right? This is a really cool hypothesis, the idea that plants put up these heavy metals into the nectaries, and over time, only the insects that are tolerant of heavy metals will pollinate you. So heavy metals are mediating pollinator preferences, and through pollinator preferences, you might end up getting reproductively isolated from your close relative of serpentine. So Mendel and Ashman, Pierre Lynn Ashman from Pennsylvania, showed this with Streptanthus polygloides, that the nectaries have high levels of nickel, and bumblebees, within seconds of visiting a plant that has high levels of nickel, that growing on serpentine soil, will not revisit that plant. It goes to a pot that's grown on normal soil. So they detect nickel almost immediately once they visit the flower, and they move on to flowers that have no nickel in them. So this is a cool idea because it really contributes to reproductive isolation mediated directly through the metal. If you are in serpentine landscapes, get in touch with me. I can give you filter paper that has a really cool colorimetric test. You can actually crush a leaf and it turns pink or reddish. If you know that there's nickel, at least 1% nickel in the tissue. So it's a really cool method that we use to find nickel hyperaccumulators in the field. But now we have a better technique. We have something called a PIAC XRF machine, which is an X-ray technology thing, handheld, and I'm hoping to send my technician over to your herbarium because you have a huge area organelle collection. Thanks to Travis and his students, there are a lot of serpentine plants. We're targeting serpentine plants. Since the pre-XRF revolution started, there have been 300 new hyperaccumulators added to the collection. So people are finding these are on herbarium specimens. You just scan a leaf for a second. It will tell you the entire elemental profile, within a second, right? So it's really fast. You can do thousands of specimens over a day. We currently have a grant from the US Department of Energy, which is lucky it came from the US Department of Energy, so it wasn't taken back. But we have this money now, and we are going to 15 herbaria. Right now, my student is at University of Washington's herbarium. We are scanning specimens. The idea is to develop a US-based metal hyperaccumulator database because these metal hyperaccumulators can be used in restoration efforts. So right now, there are this technology now that has gotten to a point where people are opening farms of metal crop plants, right? So they're using serpentine endemics that can take up high levels of heavy metals to agro-mine. Right, so this documentary that I promised Chloe that I'll send, there's a QR code for that, is about this technology. So there are plants like in the Brassicaceae, Asteraceae, and in some other families, Phyllantheraceae, that are used as a farm crop in Malaysia, Albania, South Africa, and also Greece, and soon in Brazil, where people will be growing these plants in disturbed, contaminated mine tailings to extract heavy metals, and the metals can then be extracted to make metal that can be used for industrial purposes. So the idea is you use a serpentine endemic plant that has the ability to do this. You end up with metal-rich bio ore. You treat it as a crop, so you can do this as an agricultural operation rather than using it other ways, right? So this is the idea. So nickel mining is obviously a problem. This is in Madagascar. When I was in Madagascar, this is the third largest mine of nickel, and this is in a biodiversity hotspot. There are 25 outcrops in Madagascar. 3 of them are some of the world's largest nickel mines. None of the other ones have been looked at. So it's an understudied flora, right? This is an exciting area for me to get into. But the idea is that, through agro mining, degraded serpentine landscapes, that might have a lower need to mine large mining areas, but I don't really think it's going to make a difference because the amount that plants can take up is not as high as what the industry would need. Right? Last slides, 1980 went with Robert Ondaff, 2009 when last time I visited Jasper Ridge. This is what Jasper Ridge looks like. This is what much of California looks like, where Lastenia californica and other herbaceous annuals grows. Part of this is really the aerial deposition of nitrogen, which is an understudied thread. There's some work done in UC Santa Cruz, but there's a lot more that needs to be done. What happens is when in a nitrogen low system, suddenly nitrogen is raining on you. You have non-native grasses that are coming in and basically out competing these native plants that are small, stress tolerant, they don't grow very fast. And all of the functional traits that make them perfect for a serpentine landscape is now shifting because of high levels of nutrients in the system. And that has led to the, things like the bay checker spot butterfly that relies on Plantago erecta and castanea on these outcrops. The larvae feed on these plants. These plants are getting outcompeted by grasses, and then this butterfly is now endangered. So this kind of pattern even there are examples of this type of situation in Europe as well, where high nitrogen levels of nitrogen is dropping, right? So we actually, a lot of what I do with students is when I get an exciting idea with undergrads, we actually do the literature research and somehow get the paper published in a chapter or an article. So that's a fun way to explore different areas. So people who are thinking about working at smaller schools, you can still do cool stuff with young minds and get your work out. So with that, I'm sorry I just went over a little more than I had planned on doing, but that often happens. So the cool thing, you guys know this, but like we have this idea that rock outcrops are these barren landscapes that are not exciting. There's not a lot going on. But my kind of whole kind of life mission has been to promote these outcrops as like perfect model system to ask questions, all the way from cellular level processes to ecosystem level processes. So they're not boring, they're not barren, they're really hotspots for biodiversity. In fact, you could argue that these are like the next frontiers for biodiversity research because we've done the rainforest, we've done the canopies, we've done all these other cool habitats. So now let's go look like at mine tails, which may have had 200 years of evolution to take place. And with high selection pressures you get under those settings, there could be population level differences happening on those landscapes. So with that, I'm going to end my talk. thank you for your attention. I'm really sorry that I went over 5 minutes, 10 minutes, but appreciate the opportunity.